micro injection, biolistic or the gene gun, and disarmed pathogen. And these are the two images of micro injection and as well as biolistic or the gene gun, in which directly we are inserting here, and here we are bombarding them with the microparticles bombarded with the target plant cell, etc. there, and these two things are using here in the animal cell. This will be in the case of which one the plant cell we are using also. And this is the entire process normally we are taking place at the time of the creation of a genetically modified organism. Here we can see that foreign DNA is there, and here the vector is there, and we are cutting by using the nucleases enzyme, and both the structures foreign DNA as well as the vector DNA should be cut with the same restriction enzyme to make them sticky ends. After that we are ligating by using the DNA ligase, and here we can see the recombinant DNA, and we are inserting into, this is the host, these are the methods, so that it will insert and it will absorb, and after that it will provide, and then all the genetic material will be transferred again. So this is the general activity which are taking place. Let us see what are the important processes which are taking place to creation of the materials. Also here we can see the process of recombinant DNA technology. There are different steps are there. The first step is isolation of the genetic material. Second one is cutting of the genetic material, and third one is amplification of genetic material by the polymerase chain reaction. Next one is insertion or introduction of the genetic material into the host body. Next one is getting the product of the rDNA or the gene of interest, and the last one is separation or the purification, namely the downstream process, etc. Let us see what is happening here, what are the requirements essential. Here we can see the isolation of genetic material. If we want to identify, if we have identified the gene of interest and the plasmid also, it may be of different types of the cells. And if the identified gene or the plasmid, if it is there in the plant cell or in the bacterial cell or the fungus cell, we want to isolate that gene of interest by digesting their cell wall, for which we are using lysozyme for the digestion of the bacterial cell wall, chitinase for the digestion of fungal cell wall, cellulase for the digestion of the plant cell wall. After that we are taking out the genetic material, and which will be treated with the new proteases and RNAs to remove the protein and RNA along with them, and after that treating them with the chilled ethanol to avoid all the other impurities, etc. This is the first process of isolation. After that, cutting of DNA at a specific location. As we told earlier, the cutting is done with the help of restriction enzymes. After that, we will separate them by using the gel electrophoresis. After that, we will ligate them. The plasmid with the gene of interest will be ligated by using the DNA ligases. All these activities are taking place during these steps. Cutting, separation and formation, etc. will be taking place. And at the same time, the next level is amplification of the gene of interest. If we want to get more number of copies of that recombinant DNA, we can use another technique, namely an artificial technique, namely polymerase chain reaction, and it has developed by scientist namely Carey Mullis in 1983. And an artificial medium, by providing all the requirements, we can artificially produce more number of the same copies. How it is possible? We can see the three major steps, namely denaturation, annealing and extension. In the next slide, we can see the process here also. Here, the first process is the denaturation, and this is our recombinant DNA. And this recombinant DNA, if one copy is there, we want to keep them in the particular temperature. And you know that one, they cannot tolerate the high temperature. But at the same time, we are keeping them high temperature in between 90 to 70 degrees Celsius. So that what is happening? There will be the denaturation, the separation of the double stranded into single stranded ones. That is happening by the process, namely denaturation. And after that one, we can see the two strands will be formed as each template strand, single stranded DNA. And this will be the template for the formation of the next strand, for which we are using the primers, and as well as which one of the other conditions like DNA polymerase and deoxynucleotides, etc. But the annealing is nothing but the joining. What we are doing, these primers will join with the template strand, and they will start the extension or elongation of the new strand by using the DNA polymerase and deoxynucleotides. Here we want to remember one important thing, that this is an artificial means which are taking place at a high temperature. Generally, DNA polymerase cannot tolerate high temperature. So that what we are using right now means we are using a special thermostable DNA polymerase, namely Taq polymerase, which have extracted from the Thermus aquaticus, one bacteria which are living in the hot springs or the streams, etc., high temperature of the water. So they can sustain, they can tolerate the activity, and they were doing the normal activity of polymerization also. So this is the extension also. After that, what we are getting, we will get a lot of same type of DNA fragments will be available in the case of this group of mechanism also. So the method is called as polymerase chain reaction. So we are getting a lot of billions of the copies will be available within short time also. After this one, the next process, as we told earlier, discussed earlier also, the insertion of the recombinant DNA into competent cells. Already we identified the cell, whether it is the plant or the animal or the bacteria or the fungi. By using the different methods like chemical treatment and incubation, like micro injection, like biolistic or the gene gun, or the disarmed pathogens, etc., we can insert that particular gene of interest and along with rDNA into the organism also. And we are providing other conditions essential for them. Then the next stage is obtaining the gene products. This is a very important one. We have done all these activities to get the product, like any therapeutical product, or in the case of enzymes or the alcohols, whatever the product are there, we want to take them, for which these genetically modified organism or the host organism which have prepared will be kept in the particular condition or in the culture medium. So this kind of the product what we are getting from genetically modified organism, we are calling them as recombinant proteins. The product which are getting from the heterologous organism can be called as the recombinant proteins also. And these proteins, how we will get it, obtaining the product. We are culturing these microbes in the controlled condition in the large vessels, etc. These vessels where we are culturing the genetically modified organism to get the product are called as bioreactors. These bioreactors from 100 to 1000 liters of capacity, and which is having very controlled conditions, which provide all the requirement for the growth and as well as multiplication of which one genetically modified organism also. And here the two images, types of two bioreactor image they have given. Steamed type and sparged type of bioreactors are there, which are commonly using in the commercial level for the production of the product in the commercial level also. And this bioreactor will provide all the optimal growth condition. That means the optimum temperature, optimum pH, required amount of substrate, required amount of oxygen, and the minerals and the salts, vitamins, whatever things are essential, everything will be provided by this bioreactor. And they will produce all the product, including our product of gene of interest also. And this kind of bioreactors are the bioreactors we are using for the culturing of them, thereby to get the product. The last one, how to get the product, because the culture contains all other different type of chemical products are there, from which we want to identify the product of our gene of interest. And the process consists of it's called as normally it is a downstream process. It is called as downstream processing also. This downstream process, what are the product? Like we can say the enzymes or the alcohols or antibiotics, whatever things are there, everything will be there in that particular the product system. We want to separate them. And this kind of the separation and thereby the purification of this product commonly collectively called as downstream processing. And after that one, what is happening, the product cannot be directly sent to the market, and there should be some processing will be there, in which what we can see means and here the process, downstream processing, and that means the purification and the finishing work will be different from product to product. For example, in the case of therapeutic or the product we are using in the field of treatment, it cannot be sent directly. So that what is happening, we will be treating them with some preservative as far as the food components are concerned. At the same time, if it is the medical component, what will be, it should go to the clinical trial. So thorough clinical trial should be there in the case of medical product, and at the same time, if it is in the food product, it should be added with the preservative, then only we can use it. Thank you.